Hi everyone, I am Ozkan. I'm the director for Center for Biological Physics, also professor of physics at Arizona State University. So let me share my screen and tell you about my research a little bit. So in my lab, what we really do is we study fundamental worker bees of uh, cell, which are proteins, and we manage that. We try to understand protein dynamics and also the second secret of life, which is illusory through the lens of evolution. That's really summarize what I work on. And if you're interested in my research, please email me. My email address is banoskan@asu.edu, and I'm happy to answer you and meet you in person if you want to meet and learn more about my lab research going on in my lab. So in my lab, what we really is interested is the first grid also, which is the sequence worker bees of protein. The worker bees of the cell proteins are made of amino acids, but there's a specific sequence where nature figured it out and bring this different. There's many way of bringing this twin type of amino acids, but there's a unique way you bring together, and that turns into a great function that makes one function, for example, making some enzymatic reaction, right? And other just deliver some ligands, some others just make binding and so forth, right? But we learned eventually that there is also sometimes ordered interactions, which we'll call it the sequence into some more ordered form. In that ordered form, proteins function. But when we go and analyze more, we realize is actually in cell proteins, most of the proteins are in this aquatic environment and they fluctuate, and that fluctuation, type of fluctuations gives you the function. So what we're really interested in is, for example, if you make certain mutation in that amino acid, nothing happens in the sequence. But if you make specific mutations, so then you modify that function, and that's what evolution did, right? 3 billion of this Monte Carlo step, evolution generates, starting from same ancestor, diverse functions, and this is what we really understand, how evolution builds and generate this diversification of function from like some minimal number of proteins. And for that, what we do is something quite unique. What we do is, instead of looking nowadays, it's just very easy to sequence every different species and get the same protein from E. coli to human, horse, and fish so far. But instead of looking those species, we go and predict most likely sequence of their ancestors. So it's like we asked the question 3 billion years ago, how does protein look like? And what type of mutations in the sequence enhance, sometimes diverse the function. That way we want to address how does adaptation proceed at molecular level, and what mechanism have the diverse function of mother genes are emerge. And for to that, what we do is we use molecular dynamic simulations, make predictions, which sequence is important, how the dynamics change after the mutations, and we really understand quite interesting results. We get quite interesting results about what mutations really make ancestral proteins suddenly become enhanced and a superb function, for example. And to really understand this, we start also designing novel proteins and novel enzymes. Is you say, what I cannot create, I do not understand. So when we understand what's going on in molecular level, we start making new proteins to really check what we're learning. And this was a statement that with Richard Feynman. So we do and start designing enzymes. So to do this, we leverage sequence function inputs from ancestral studies to use protein dynamics and design proteins with novel functions. The second thing is, in human also, like my proteins are completely different than your proteins because we have genetic mutations in our DNA, right? We call this single nucleotide changes, single nucleotide variants, SNVs. It can be in protein coding region of our protein, or it can be in non-coding region of protein. Non-coding region is quite complicated, but if it is non-coding region, some mutations, some single amino acid mutations doesn't change the 3 codon nucleotide. We still have the same amino acid, but sometimes the change in one single nucleotide gives this triplet a different identity. Some of them, like stop codon, we call that nonsense. Some of them can be a mutation in your protein, like you have a protein which make you a very good person, like a good athlete sometimes, or sometimes susceptible to disease. So we also in our lab study also apply this type of learn to predict what type of missense mutation can lead to disease, and can we understand and find disease mechanism and possible cure for those diseases. With that, I'm looking forward to hearing from you.
And all of our research is biophysics, so it's the combination of applying physical principles to biological systems. So you can take a lot of this work in really any of the directions that you want to go. And I'd always hope that whatever I'm doing becomes legacy work for some of the people that come onto the program afterwards.
Right now I'm working on trying to figure out how evolution works. In order to actually build some antibiotics which can help us fight against the bacteria, we need to understand how they evolved. I think it is the thirst to answer these type of questions which drives me, and I think that's the same reason why I'm working with Dr. Ozkan, because she lets me work the way I work.
If we can understand this, we can design better drugs, we can prevent diseases, and we can predict based on genome sequences like what type of diseases a person can get, which is very popular with this precision decision at the moment. Impact will be obviously human health.